So our last speaker for the session is Nadav Kislev from Daphna Benayahu's lab.
So hi everyone, my name is Nadav from Daphna Benayahu's lab. First of all, thank you for the organizer for giving me the opportunity to present my work here today. Today I'm going to talk about SVEP1, its role and therapeutic potential in adipose tissue. So we'll focus on obesity in adipose tissue. Obesity is considered by many as a 21st century epidemic. It is characterized by disproportionate body weight for height with excessive accumulation of adipose tissue that is usually accompanied by chronic, systemic, mild inflammation. Now, obesity was previously shown to play a role in the development of other pathological conditions, of course, such as diabetes type 2, cardiovascular diseases, several types of cancer, and some other severe pathological conditions. Now, adipose tissue lies at the center of obesity pathogenesis. During obesity, it goes through immense remodeling processes, and excessive caloric intake usually causes the adipocytes to expand substantially. Now, those hypertrophic adipocytes trigger alterations in the differentiation capacity of the tissue that limit and impair adipogenesis, which is the differentiation of mesenchymal cells to adipocytes. Those hypertrophic adipocytes also secrete pro-inflammatory factors that can, of course, further change the dynamics in the tissue, lead to infiltration of pro-inflammatory cells, change the immune phenotype of those cells, and of course alter the cytokine milo as well. Another characteristic of the dysfunctional adipose tissue is local hypoxia, which is caused by insufficient perfusion to those hypertrophic adipocytes that again further causing fibrosis and inflammation. Now altogether, these processes lead to impaired glucose metabolism and lipid metabolism and change the systemic metabolism in the body. Now in our lab we study SVEP1. SVEP1 is a 370 kilodalton protein. That's an ECM protein, a pretty big one. Of course it is involved in cell adhesion. It is expressed in mesenchymal cells and tissue, and it was previously shown to play a role in several metabolic diseases, like atherosclerosis, coronary artery disease, and also obesity and diabetes as well. Now, in adipose tissue, SVEP1 is highly expressed in adipocyte. It is upregulated in adipogenesis and obesity, where it gets secreted to the ECM and interacts with immune cells, but also can act on the adipocytes themselves. As you can see here, SVEP1 is highly expressed in both the adipocyte, this is an epididymal adipose tissue, and mesenchymal cells, and it is upregulated in high-fat diet or diet-induced obese models. When we do a knockout, a heterozygous knockout of SVEP1, we see that it can also affect the systemic glucose homeostasis with changes in fasting glucose tolerance, and also insulin tolerance as well, with the knockout mice presenting more tolerance to insulin and glucose as well. Now, SVEP1 has a known primary receptor, which is called integrin alpha-9 beta-1. It is a relatively new addition to the integrin family. Of course, it is expressed on the transmembrane, where it acts as a cell adhesion molecule. But not only, it also promotes proliferation and inflammation as well. Now, it is expressed on many cell types, including myeloid cells, where it interacts with its many ECM ligands. In myeloid cells, integrin alpha-9 beta-1 was previously shown to play a role in osteoporosis and some other pathological conditions through this pathway that is shown here. And again, where it promoted proliferation, inflammation, migration, and adhesion. We started off with studying integrin alpha-9 beta-1 expression in adipose tissue, and we saw that it is mainly expressed in myeloid cells, and more specifically in macrophages. And here in this staining, you can see that it is highly co-localized with F480 in both high-fat diet and low-fat mice. And when we studied the different subpopulations in adipose tissue macrophages, we saw that integrin is quite ubiquitously expressed across those subpopulations, with slight upregulation in the pro-inflammatory infiltrated macrophages subpopulation. Now, we know that SVEP1 has a known binding site to integrin alpha-9, beta-1, that can act independently and separately as a peptide with the potential to inhibit the integrin's pathway, of course, making it a potential and potent therapeutic target. So we used this peptide and started study whether the peptide can act on, first of all, on cells in vitro. So we started off using gross cells. This is how they express the integrin. We performed some dose and time response assay, knocked down integrin, and saw that the peptide, the SVEP1 based peptide, is quite specific to the integrin. We then measured whether the SVEP1 based peptide can inhibit the integrin's pathway. We started off with the bread and butter of integrin, which are migration and adhesion, where we saw reduction in both adhesiveness and migration of the cells when they were co-incubated with the SVEP1 based peptide. We then moved on to study its effect on inflammation. We induced inflammation using LPS, and co-incubation of the cells with LPS and the peptide reduced both nitrite levels and also the phosphorylated ERK fraction, which is one of the downstream effectors of the pathway. It also lowered the cytokine levels as well. So we knew that it can affect in vitro macrophage-like cells, but we wanted to assess its potential in the adipose tissue. So first of all, we used ex vivo tissues. We extracted the epididymal adipose tissue, the visceral adipose tissue from obese mice, used them as explants, where we activated those explants with LPS and incubated them with the peptide, and then studied the immune population and bulk analysis. As you can see here, the peptide lowered TNF-alpha levels in an intracellular cytokine flow cytometry analysis in both immune cells as a whole, but also specifically in macrophages. And we saw a reduction in some cytokines as well in a bulk assay that we did to the tissues. We then moved on to an in vivo model where we injected the peptide straight to epididymal visceral adipose tissue of high-fat, diet-fed mice. So basically, we used diet-induced obese mice. And after 12 weeks, we injected them saline with and without the peptide 3 separate times. And then the first analysis that we did is measuring the weight difference. And we saw that the peptide-treated group stopped gaining weight compared to the vehicle group. We then moved on to assess its effect on glucose. And we saw that the animals that were treated with the peptide were more glucose tolerant than the vehicle-treated ones. Now, we know that the peptide affects inflammation, and adipose tissue inflammation is quite important in adipose tissue dysfunction. So we then moved on to study the effect of the peptide on adipose tissue macrophages infiltration, where we saw lower levels of macrophages in the tissue. And not only that, we also study the specific characteristic of macrophages in the tissue. One of the key characteristics of the dysfunctional adipose tissue are crown-like structures. These are aggregates of macrophages that surrounds usually necrotic adipocytes. These cells become really pro-inflammatory and dysfunctional, and we use them to assess the amount of dysfunction in the tissue. So as you can see here, when we used the peptide, we had less crown-like structures, and the cell expressed more 206, which means that they also changed their phenotype. And this is in vivo. So to conclude, integrin was quite ubiquitously expressed in adipose tissue macrophages subpopulation in both obese and lean mice. A peptide that was based on SVEP1's binding sequence bound to raw cells and effectively inhibited inflammation in the cell. The peptide also reduced the activation of adipose tissue explants, and local injections of the peptide to visceral adipose tissue exhibited its potential as a therapeutic agent against adipose tissue dysfunction. I would like to thank my lab and Professor Daphna Benayahu, and of course the center for giving me the scholarship and the opportunity to talk here today, and thank you for listening.
Questions? You ever talk about like a different pandemic, huh?
Yeah, this is a kind of out of scope, and
So
sort of a
how
different.
can you elaborate on then, what happens when the adipose tissue is inflamed? I mean, what
Of course. So the dysfunctional adipose tissue or adipose tissue in most obese patients, usually what we see is these hypertrophic adipocytes. They secrete a lot of pro-inflammatory factors that further cause a lot of immune cells to infiltrate the tissue. So in healthy subjects, we see that macrophages are about like 5% of the cells, but in really like obese and unhealthy subjects, they can get up to 50% of the cells. And then what happens? They secrete pro-inflammatory factors, and of course they further cause like other damage. Yeah, and then this is like a vicious cycle of inflammation. And this inflammation is not only bound to the adipose tissue, but of course it affects it has a systemic effect. And this is one of the reasons why obesity is such a comorbidity, and so, yeah.
Okay, any more questions? Otherwise, we'll wrap the session up, and thank all the speakers. And, okay, so in another, 15 minutes? Or another few minutes, we have closing remarks by Ty, so we'll stay here.
Good afternoon. I see that everyone is eagerly waiting for my closing remarks. These will be brief. Thank you to all the speakers, all the participants, to Yael who made this conference possible. And I'll see all of you next year in the next conference. Thank you.